I'm going to be teaching on bioethics, specifically on creation of life ethics. Disclaimer. As this presentation deals with a subjective and highly charged topic, I've made an effort to include only verifiable facts and common conclusions, unless otherwise specified. That said, any unacknowledged or acknowledged opinion or bias appearing in this presentation should be assumed to belong solely to the creator, which would be me. So, the reason for this is that bioethics as a whole is not hard and fast. It is not facts. It is not something that you can pin down. All of these topics are subject to debate because they are debatable. They are not something where you can measure it. They are not something that you can just take a reading of and say, oh, this is what is, this is what, because it is what, this deals with what should be rather than what is. So, as a result of that, much of my presentation is going to be involved in providing background information about what is, so that you can be, so that we can be informed in discussing what should be. So, to begin, passing out handouts. Please take one, look at it, look over the questions. You don't need to actually answer them. I'd appreciate it if you would look at them and think about them. You could write down your answers if you'd like to, but that's not what's the most important. The most important is that you just consider them. And I'll be getting through the majority of them, but as I said, some of the questions are just for you to think about and consider. So, what is bioethics? So, who can give me a definition of bioethics? If I asked you to define it, what would you say?
The ethical issues surrounding things in biology, doing things in biology.
For your definition?
Kind of like morals relating to science of like what you should do, what's like socially acceptable.
Right. Anything else? Yes. So, not only is bioethics itself hard to pin down, the actual ethics, but even the definition can be somewhat challenging. The definitions that I found, so the original usage of the word came in 1970 by biochemist Van Rensselaer Padre Blanc II, and he said, the bioethics is biology combined with diverse humanistic knowledge, forging a science that sets a system of medical and environmental priorities for acceptable survival. I doubt to his experience, but I find that this is less than useful for what I'm trying to do. So, I shall instead rely upon the definition provided to me by dictionary.com and several other dictionaries. Dictionary.com provided a very succinct explanation, so I'll be sharing that with you. And it is the study of ethical problems arising from biological research and its applications in such fields as organ transplantation, genetic engineering, or artificial insemination. Those last three categories are just several of the many examples of the sort of fields that this topic deals with on a regular basis. Excuse me. So, again, this is an extremely broad topic, so I'll be narrowing it down and focusing on a more specific topic, but we'll be getting to that in a little bit. So, next, major issues. So, tell me, what are some major bioethical issues that you know of? What are the big controversies? Dylan?
I know a recent one was where stem cells came from. Well, some of them came from, or like in the past, like most of it, or yeah, the most common stem cells were embryonic, but now they can be taken from other places, I guess.
Okay. That's a really good one. Other topics?
Genetic engineering in foods, so who should be responsible for deciding whether we should have genetically modified plants in the grocery store, and things like that.
Right.
Someone who's in a coma and is living off a machine.
Yes. So, extended living should we keep people who need life support to live? Should we make that choice to keep them alive?
Mm-hmm.
So, of the topics that I chose as examples, you covered several of the big ones. Stem cell research, again, the choice to take to research on embryonic stem cells is a big, that was a very big debate, and it's still not solved, even though there are now other sources that stem cells can be taken from. Transhumanism, the modification of human genes in order to create effects on people, either to fix genetic defects or, in some cases, to just create an entirely new effect in order to improve the human race, or so the advocates would say. Assisted suicide. Should people be given the right to end their lives if they so desire? Creation of life, which I'll be focusing on for the majority of this presentation, and gene therapy. Again, that plays into part of the transhumanism with modifying genes. And, as I said earlier, this is not hard and fast, so I will be doing my best not to give opinions, or rather, I will not be doing my best not to express my opinions as facts, because I can assure you that they aren't. So, instead, I'm going to start with dealing with things that are facts, so that you have some background to discuss and think about the ethical implications of life. So, for the creation of life, the two big methods that I looked into are cloning, and which has been around for quite some time, and cell synthesis, which is relatively new. So, cloning. So, what everyone thinks of as cloning, the first thing that comes to mind when you hear the word cloning, what? Star Wars.
Yes.
Cats.
Star Wars.
It's all in.
And follow the sheep. So cloning in society in general, in the majority of its uses, has come to embody one aspect of cloning, which is more accurately referred to as reproductive cloning. So reproductive cloning is the formation of, or rather, the duplication of a pre-existing organism's genetic code in a new organism to create an exact genetic duplicate. There is also DNA cloning, which is what we have done in the past with bacteria to create antibiotic-resistant E. coli, for example, in which you only take a small section of the genetic code of an organism and transfer that into another organism to create an effect. Reproductive cloning, on the other hand, creates an entirely new organism, and this is typically done through the process known as somatic cell nuclear transfer. So, how this works. So this will be representing the DNA, right? So you have a cell from a live organism that is fully grown, fully developed. So this has its own DNA in it. In order to excuse me, in order to create a clone, you need to first remove this DNA from the cell, typically destroying the somatic cell in the process. So then you have the new DNA to work with. So now that we have this DNA, what you need, DNA on its own can't grow. So the typical process that's used to create a new organism is to take an egg cell from another organism and then remove its nuclear information, its genetic code, and implant the genetic code from the original organism that you want to duplicate into it. So at this point, the DNA from the organism that has already been grown, that is already alive, will be determining the processes of the new cell, right? So then, for mammals or other such creatures, this egg cell needs to be injected into the uterus of a suitable mother, because eggs sitting outside the uterus have a distinct tendency to die, and that's counterproductive. So the egg cell is then injected back into the ovary where it can grow and live, which allows for the, as I said, which allows it to grow, which in this case is the replication of the cell. And interestingly enough, in spite of the fact that all of these are the genes and chromosomes from the somatic cell, right? So in a full-grown somatic cell, various genes will be turned off so they are no longer functional. For example, in a liver cell, you don't need the same proteins to be functional as you do in a nerve cell. You don't need to be able to move the same compounds. You don't need to be able to create the same effects. So these genes will be turned off. But in spite of that, when you put the new genes into when you insert the genes into the cytoplasm of the egg cell, if the technique is successful, that won't matter,